All right, welcome to this talk on intersubject correlation and naturalistic stimuli. I'm your captain today, Pete Molfese, and I know you've seen me many times. This is my, fortunately or tragically, last presentation of the summer series this time. Helps if something moves. Okay, I am part of the fMRI core facility and benefit from many people smarter than I am around me to give me ideas for some of these studies. And if you find intersubject correlation to be fascinating or interesting and you want to learn more, keep in mind that this is a less than 1 hour talk. There is this workshop online that I ran in 2021. It's even got it in the URL there, on naturalistic stimuli and individual differences. We had 12 presenters, 2 in the intramural program and then 10 in the extramural program. It is online for everyone to watch. You can scan the QR code, you can type in the URL, but it is roughly 9 hours of people presenting their work on doing naturalistic stimuli, ISC, all of that. It's a fantastic workshop. I encourage everyone to watch it. And we are somewhere in the 5,500 people have streamed it at this point. So that should give you an idea for, since 2021, how popular ISC has become. And I want to kind of, as I started writing this talk, I was like, okay, where does this fit in? And I came up with many answers. And so this is just going to be my interpretation. But we kind of started off with a history of MRI and brain imaging in general. And then we kind of went over here to this other side and we gave you 2 talks that were like the basic fMRI study and then apnea pre-processing and how to like look at your data and really see what's going on. We started this 3rd branch that went into how to get better data, and better could be defined either as multi-echo or high resolution. And then along with that, you also learned something about, hopefully, the physics, the physiology and confounds, and then also noise and artifacts that come with fMRI data, and hopefully how to mitigate some of those. Peter gave a talk last week on controversies in fMRI. And then there were these 3 talks that were given, 2 by me, 1 by Catherine Walsh, on other kinds of data that can go with your fMRI data, whether that's multimodal, whether that's a type of multimodal EEG, or whether that's relating back to behavior, which I think is in theory why we all do brain imaging, because we care about human behavior and we want to know how the brain fits in. And then as I was making these slides for today's talk, I realized that in past years, we've had a couple of talks that were sort of key. And so one of those is we never really had this year an fMRI design and optimization talk. And we also didn't have a resting state MRI talk. And the reason I bring these up is that today I'm going to talk about those because ISC, you'll find, kind of branches off of those very naturally. And so today we're all here for intersubject correlation. Let me give the quick recap for anyone who's new or anyone who just likes seeing figures of fMRI state designs. So you can imagine in any fMRI study, you can have a time series and you might have a series of blocks of 2 conditions. Say you have an arithmetic block and you have a reading block, and then you're going to contrast how the brain activates doing these 2 different tasks by doing a subtraction. You can also have an event-related design where we have more discrete events. So instead of 40-second blocks, we might have like trials that take a second or even less, and we get nice robust activations. And if we were to look at that event-related design in the way that AFNI and other softwares represent it, it kind of looks like this. I did the first 5 of this correctly, and then I got distracted. But you can kind of see like the first 2 conditions represented in black show up as on the top series, and then the next 3 show up in the bottom series. And the reason I bring this up is that in traditional MRI design, if we are taking our analysis of either blocks or event related, we go from whatever our design is, whatever is on the screen, to brain by first convolving this design with a hemodynamic response function, whether that's a gamma function, whether that's a modified gamma function, in order to represent how the brain activation would actually look after seeing an event. And then at the most simplest basic level, if you just correlate every voxel in the brain with this time series, anything that is correlated above particular p-value gives you activation in the brain. So those voxels represented here in yellow are positively correlated with your design. Those voxels shown in blue are negatively correlated with your design. fMRI in a nutshell for basic design. And fMRI has been around since the early 90s. And only a few short years after fMRI came out with task, with like things like finger tapping, this group came out and they discovered that there's also just the brain at rest. You just put someone inside an MRI scanner and you say, chill out, do whatever you want. The brain does something. In fact, it probably does what most of us are doing most of the time of our lives, sitting on the metro, things like that. So resting state MRI has no task. Usually you're just given the instructions of either keep your eyes open, like it was crosshair, close your eyes, relax, don't fall asleep, things like that. And Biswell showed in 1995 that if you just take one seed in motor cortex along this time series of resting state and you correlate it with the rest of the brain, you get sort of contralateral activation of the brain. So you have these regions of the brain that are naturally correlated with other regions of the brain. And you can analyze this data in a number of ways. You can do things like functional connectivity, which is just a fancy way of saying correlation from point to point. And you can make assumptions or make statements about how these networks may or may not interact. And if you're interested in this more, even though it got cut off a little bit here on the screen, Katie Chang gave an awesome talk on resting state MRI back in 2019. It's online on the website. It's also on YouTube. I would highly recommend you check it out because she spends an entire hour talking about this. In the next two slides I'm going to show you, but I just want to give you kind of this baseline on how we got to ISC. So if we sort of continue on the theme of, well, you now know what task is and you kind of know what rest is, like task, we have our design and we're correlating the brain activation to that design, and we get activation among certain voxels in the brain. In rest, we just have a time series of what the brain is doing while you're laying there, looking at something, not looking at something. And then you can correlate that time series with any number of things. And when resting state first came out, it was all the rage because it's like, well, you don't even have to do a task. Like you can just put people in the scanner. It's like, oh, like, you know, we're still figuring other things out, like just have people chill out in the scanner for a while. And then people kind of came along and said, well, how long should we have people do resting state? And most people were like, 5 minutes is great. Like 5 minutes sounds like a good round number. And then there was this paper that came out that I'll show here in a second that essentially said, well, resting state connectivity gets more stable the longer you do it. So the question then becomes, is it just 5 minutes or is it like, should I just do resting state for all the time that I have? And I know that Carlo's on here for diffusion tensor imaging, and he's going to talk about this later, but typically when people say, well, how much diffusion should I do? People say, well, how much time do you have? And in resting state, it's sort of that same kind of thing. It's like the longer that you add resting state, the sort of better your interclass correlation becomes. And there was this awesome paper by Rasmus Bern that came out in 2013, which I swear I have saved on my computer at least 8 times, because every time someone says, how long should my resting state be? I go to send them this paper and I can't remember where I put it the last time. I should probably just bookmark it. Yeah. And so what Rasmus showed is that, you know, your interclass correlation gets 20% better if you're at 12 minutes or more, and that your functional connectivity measures are much more stable as soon as you go above 12 minutes. But there are also suggestions that you should be doing resting state for even longer than 12 minutes. You should be doing it for, you know, as long as you need in order to get to the interclass correlation that you want, which makes a lot of sense if you've ever taken a stats class with any kind of power analysis. Like, you should be doing the thing that you need to do to get the signal, to get the significant results that you want. Resting state's also particularly cool because the brain is full of all of these interesting intrinsic networks. And so here, what they did was they put a seed in sensory motor cortex, and then they just took the seed time series and correlated that with the rest of the brain. And what they found was that this seed to whole brain can pull out the rest of the sensory motor network, so to speak. And that if you put other seeds in the brain, you can pull out, say, 7 or so major brain networks by putting a seed somewhere in the brain. And they're kind of shown here. And yeah, these networks are very reliable, but there are some differences in chemical groups, and this literature is vast and wide. And if you're interested in more of this, I definitely would say check out both this original paper on the restless brain, but also Katie Jean's talk from 2018 or 2019. So that's a lot of, I have one seed in the brain, I'm correlating that seed to the rest of the brain. The other thing that we can do really easily is we can take the brain and we can have multiple seeds, and we can just say, okay, like what happens if I look at 4 regions? How do those 4 regions correlate with one another? And I can do that pretty straightforward. I can just take those time series and I can just do a temporal correlation. And I can say, okay, these temporal correlations or synchronizations in the brain happen. And I can study this both at rest or even during the task and potentially get really important information out of it. For example, you could do functional connectivity with multiple seeds. And say you have 3 participants, they may have different representations of what this correlation matrix among these 4 seeds looks like for each of these participants. And this is sort of the underlying theme of Emily Finn's paper, which those people in the room can kind of see in the upper right corner, on fingerprinting that came out several years ago now, which really said, hey, based on resting state connectivity or even task connectivity, I can tell you who is being scanned at the moment. So if you have 2 runs of the same task on someone, can you say, this functional connectivity definitely belongs to Bob, and this resting state connectivity or task connectivity definitely comes from Susan, so on and so forth. For anyone who wants more information on functional connectivity, it's a great paper to check out with John Gore as the senior author. And the reason that I've given this background is resting state is a powerful tool. I don't want to discount how powerful of a tool it is, but it turns out that there are benefits to having designs, and it turns out that there are benefits to people doing something with the scanner. And so again, functional connectivity, we have rest where you're just looking at a fixation, or we might have something like Inscapes that Tammy Vanderwall did from Yale in 2017. Which Inscapes, if I just say fancy screensaver, that doesn't really do it justice, but that's what it always feels like to me. Or you could watch a movie like Ocean's 11. And so if you kind of look at this, you can see that what they tried to do here was predict which subject was which based on the stimuli. And what they found was that resting state, you could definitely do subject fingerprinting, but Inscapes was better at doing subject fingerprinting. And showing Ocean's 11 was even better at doing fingerprinting. And so you can kind of get where I'm going with this, which is like, hey, having a little bit of drive to resting state scans like Inscape is better than just like laying there looking at crosshair, but you can do a lot with movies in particular. Okay, so another recap. I know I'm going to do several of these. And that's because all of these are incredibly rich literatures that all have fantastic merit. So traditional designs are great for tasks. What's the brain during reading? What's the brain look like doing math? How do group A and B differ doing the same kind of task? We know that the brain activation, because we have a design, is the brain activation related to, so we need to design your study, right? Related to the task at hand. Resting state, similarly, can be super useful. You can look at different networks in the brains, how those brain networks change within subject, within group. You can also do functional connectivity, and you can look at sort of even fingerprinting. Here's that fingerprinting reference for anyone who missed it before. And we get to this like interaction of like, okay, there's a lot you can do with tasks, there's a lot you can do with rest, there's a lot you can do with functional connectivity, and as Tammy showed, there's a lot that you can do with movies in terms of fingerprinting. And then it kind of comes up with this like, well, if you can do a lot with driven stimuli, movies can do that in terms of stabilization of functional connectivity. What else can they do? It turns out that movies are great stimuli. Hollywood is very good at manipulating our emotions and our internal states. And I watched a rom-com the other day, and you know, my partner didn't find it nearly as fun as I did, but yeah, I'm sentimental and like old movies. But movies bring about a lot. And so this very classic, although that makes it sound old when I get the shivers, paper by Uri Hassan in 2004, published in Science, had participants watch The Good, the Bad, and the Ugly while they were in the scanner. And if you haven't seen The Good, the Bad, and the Ugly, it's definitely a movie you should watch. You are welcome to watch your phone at the same time, but like, you should kind of know the general theme of The Good and Bad and just watch a YouTube clip. So participants watched 30 minutes of the movie, and then they picked out 2 ROIs. So kind of a fusiform face area, an area of the brain that is known to be sensitive to faces, and another area in the brain that was thought to be sensitive to buildings, structures, bricks, things like that. And they ran intersubject correlation for the time course of each of those ROIs. And what they found was these time series are highly consistent across subjects. And I'm always cautious of getting too far away from the owl. But if you see all of these times where we get kind of to the peak or to the valley, it's kind of highlighted in green or red, those are moments in the time series where people were highly correlated with each other. And if you were to take that point in the time series, go back roughly your hemodynamic response function delay, and look at what was happening in the movie, people were seeing faces or they were still seeing buildings. And so they have essentially found tuned areas that people synchronized on to be like, aha, faces, everyone sees the face, everyone's fusiform face area kicks in, so on and so forth. So really, really cool study idea, because now I'm not asking them to do anything other than attend to a movie. And I don't know if anyone's ever been at a restaurant and trying to talk to other people, and then they have like a TV over there. Like TVs are really good at like magnifying everyone's attention. So at the heart of it, intersubject correlation is pretty straightforward. It is just the correlation of each voxel pairwise across subjects. So when 2 people watch the same movie in the scanner, many parts of their brain may synchronize. And this is just correlating the same voxel in participants A and B. And so you have, say, voxel A, I, J, and K, and voxel B, I, J, K. And we just take those 2 voxels and we correlate them and say, hey, were they correlated over the span of this task or over the span of this block? And then we can go from there. This is incredibly easy to do in AFNI, because even before we even thought that this would be a thing, AFNI had a tool in it called 3D T-Correlate that would allow you to time series correlate whole brain volumes. And then a few years ago, gosh, 6 or 7 years ago now, Gong Chen wrote just 3D ISC for doing group analysis with ISC to get around a lot of the statistical assumptions of paired dependency in design. The other thing that I really like about ISC is that you can make ISC an analysis about what we have in common. And so, it's like what combines us, not what divides us, common theme. And that is also something that you can say like, hey, like, can I do this with any group of participants that might be interesting in and of itself? We want to talk about why this movie thing was kind of critical, because it all comes down to the assumptions of your design. And so if I kind of split things into 2 worlds of things that have a design, like GLMs, and then things that don't have a design, becomes pretty obvious. I have a lot of assumptions going into a traditional fMRI study on the stimuli and the task. So I can put stimuli on a slide, but are those stimuli valid? Are those stimuli going to be good? Is it something about those stimuli that is going to drive a brain response that isn't the thing that I think it is? So is the luminance of your stimuli different? Have you introduced subtle variation in your task that is going to drive differences that you didn't expect and that you won't be able to account for statistically? So there are people who now put into their regression things like the luminance or the auditory loudness, things like that inside of their task in order to get around some of these assumptions or sort of account for some of these assumptions. Resting state functional connectivity has fewer assumptions, but it does still ask this question of what really drives this activity. So, and is it the window length, as I showed before with both Biswal and Rasmus Bern? Is it something about how we collected this data that is kind of making things happen? Are people like falling asleep? Are people like thinking about their grocery list? Are people thinking about what they're going to do with the money that you paid them to be in this MRI study? Are people thinking about like a movie that they just saw, things like that? Whereas intersubject correlation doesn't have those assumptions, right? Like everyone's seeing the same stimuli. And what we're asking is, are their brains synchronizing or not synchronizing? I will also toss in that there is another analysis that I'm not going to talk about today, called intersubject functional connectivity, where you can have one voxel in person A's brain and see how that predicts voxels in person B's brain. And you could see that the sky's the limit on that, including things like gambling tasks or hyperscanning, where you have people interacting, and then you could see, like, can we predict people's behavior or people's brain and behavior from something that happened? Yeah. Pros and cons of ISC. Well, let's start with pros. It's the most driven brain responses, then better than the rest, sometimes arguably better than task, because it's more naturalistic and the stimuli, in theory, approximate real life a little bit better. And we've seen everything from like very creative, like video games being played in the scanner, where sort of experimenters manipulate that Mario's not going to make that jump, and then you're going to get Mario falling into the crevice, and then you're going to get the Mario sad sound, things like that. And there's a really growing track record of ISC's use in categorizing individuals based on those states and traits. And I'll talk about that in a couple more slides. But it's not perfect, right? So at the most basic level, ISC is a pairwise measurement. And what I mean by that is that up until recently, it's fairly hard to get to individual differences, because everything that we do at the base of our analysis is person A correlated with person B, or like the combination of everyone correlated with everyone else. And so it's hard to say, like, hey, I know that this person has this particular behavioral measure, and how do I relate that in? And it can be tricky to go back and freeze a particular moment, because if, you know, in the original Uri Hassan paper, they were able to say, like, hey, faces were on the screen, buildings were on the screen, and that's awesome. But are there more subtle nuances related to what's going on in the movie? And there have been a lot of like one-shot movies that have come up recently, where instead of having cuts, you just have, like, you know, imagine a room full of dominoes, and you start the dominoes, and like dominoes meets mousetrap, and like you get to see all of these things interact with just like one camera view going over the entire room. It's like there's a lot going on in those. What are you going to pull out that you weren't expecting to pull out? And of course, there are just general problems with across-subject analyses. And there's this paper by Fernando, who's sitting in the front row, that I will say I read last night, and it is a joy to read. And it does raise a lot of questions, not just on ISC, but a lot of questions on just like across subjects, combining into group analyses, and how we do like cross-subject registration and things like that. So I'm going to put it here so that people can check it out later. But let me go ahead and address one of those cons for what if I do care about individual differences? Well, fortunately, there is a method for that, just like there's an app. There is representational similarity analysis, RSA. I'm going to give the short overview today. It goes much deeper. Not everyone loves this method, but it is growing in popularity, and it does, you know, much like the rest of what we do in brain imaging, give us results that are pretty believable. So similar, we'll say the assumption is similar behavior should also mean similar neural responses, right? Like you have even if you were black box, what goes in drives behavior. Generally, you'd imagine that the gears inside work somewhat similarly. And so what we have is this figure from Emily Finn's 2020 idiosynchrony paper. Here at the bottom level, we have our individual subjects, and we construct a similarity matrix of behavior, that's our middle level, and a similarity matrix of our brain responses, that's our top level. And once we have those two similarity matrices, we can just correlate them or do a mantle test on the two matrices and get fairly useful things out in response. And you might be like, okay, well, got a correlation of correlations, like you're running stats on stats. How do we interpret those? And I'm glad you asked, because I made a slide for that too. Emily, also in this 2020 paper, went further to say, well, you can take these ISC sort of matrices, and you can sort the rows and the columns in different ways based on their behavioral scores. And so you have two sort of very fundamental archetypes that come out of this. One is this idea of nearest neighbors. And so this essentially says that subjects similar in behavior will be similar in brain relative to each other. So if you have two people who are really good at a task, they'll be highly correlated with each other. If you have two people who are really bad at a task, they'll be highly correlated with each other. And then anyone who's in the middle will be highly correlated with each other. So it's essentially, you are most correlated with people who are about the same level of proficiency as you on that task. The other model that she proposed was this Anna K model, which comes from the first line of Leo Tolstoy's book, Anna Karenina, says that all happy families are alike. Each unhappy family is unhappy in their own way. And so here, the major subtle difference in wording is just instead of ranking things relative to each other, we are now ranking things as an absolute, not relative, as we move up and down the scale. And so you can think of this as, instead of being most correlated with the people that you're like, people who are very proficient at something, people who synchronize together at something, or even scenes that are highly correlated with each other are going to be similar. So going back to the Uri Hassan example, all the face scenes, those are highly correlated with each other. And if I was to take chess players and have them watch a chess match, the highly competitive chess players would likely all correlate together. And then the people who are very not great at a task, or even people in the middle, would likely not be correlated with each other because they'd all be doing their own thing. So they'd be uncorrelated with each other. And yeah, and it's this idea that you hit a particular level of proficiency. And once you hit that level of proficiency in whatever you're trying to measure, then people start to show high correlation. So I'm going to give several examples of this. Okay. And so ISC using RSA works quite well. It's sensitive to the state of the subject, can pick up on things like the features of the stimulus, manipulations of attentions or prior belief, or even like mood contagion, any kind of priming, you can do that with ISC. It's great for behavioral ratings of stimuli, can also drive ISC, and you can ask people later on what their interpretations of the stimuli are and like relate that back to the ISC. So there's a lot of like interaction of behavior pre and post that can be picked up on. And in addition to picking up on someone's state in the moment, ISC has also been shown to be very sensitive to traits of a participant. So people who are, say, high on paranoia, this is another one of Emily Finn's papers, or people who look at stimuli very holistically versus more analytically, people who have depression, or more and more and more. So just to give some examples, here's Emily's 2018 Nature Comms paper on trait paranoia. And what she did was she had participants listen to a fairly ambiguous story with some possible suspicious events. So it's the story about a physician who gets a letter from an old colleague that says, hey, like I'm doing medical work in this remote village. It would be great if you could come here and help. And so the physician's like, oh yeah, of course, I will fly on down there. And when he gets there, can't find the physician. The people are all very friendly, but then there's like little moments where they're like, oh, you shouldn't go there. And oh, like, yeah, the physician was just here. Here's his tent. And then like the last entry in the physician's journal is missing. It's like months ago. So there's like little ambiguities that kind of maybe pique your interest. If this was a choose your own adventure story, I think about half of the people in this room would be like, where's the get back on the plane and go home entry in this? And so Emily took this story and she ran intersubject correlation and found that, as predicted, lots of language areas are highly correlated as people listen to this auditory story in the scanner. I'll also say she had a visual stimuli, but it was just a static image of kind of like a village in a rainforest, which going through this task definitely made the story resonate more. So ISC pulling out language areas very nicely. And then she went and correlated people who were high and low on paranoia. And it pulled out kind of two specific areas. It was the left temporal pole and the right medial PFC. And so it turns out that if you then take these two regions that have been attributed to paranoia and story processing in neurosynth, and you just take your similarity matrices and you correlate them to the paranoia behavior, you can see that they're very positively correlated. So It's kind of cool. We have a trait of a participant being paranoid, and then based on their connectivity, we're starting to see like, hey, this does relate to them. Importantly, although I'm not going to go into it, Emily also had participants do a free recall narration at the end of the story where they would talk about the story and then answer some multiple choice questions. And then she did further analyses where she would relate what people said back to how they processed imagery in the story and found even more results. But I'll leave that up to you all for homework. Sort of inspired by this, Dave Gingras, Emily Fenn, and I did a study where we had adolescents reading and listening to long narratives inside the scanner. And so we had a design that looked like this. So you were either reading a story for 60 seconds, word by word, or you were listening to a story for 60 seconds, word by word, obviously. And then it would toggle back and forth between reading the story and listening to the story. And people listened to 4 different stories. So we had a pretty substantive amount of data on these people. And again, if we look at just the intersubject correlation of people doing this task, we again see nice correlation in language areas, particularly speech and language areas, including like angular gyrus and temporal lobes. And then Dave also pulled out several ROIs and like, for instance, inferior SDG, and was able to show that just like in the Anna K model, as people get better, or as people have sort of better behavioral reading scores, they tend to be more highly correlated. And then if you compare sort of our 3 groups, doing air quotes, so the red are high proficient readers on their behavioral scores correlated with other high proficient readers. The yellow are high proficient readers correlated with low proficiency readers, and then the purple are low proficiency readers correlated with other low proficiency readers. You can see that in not just this brain area, but many areas, we can differentiate the groups based on typical reading areas. This is particularly cool because in your average reading study, usually you have people reading like a single word at a time, and it pops up on the screen and they're like, I know what that word is, and they go on to the next word and they go on to the next word, and there isn't this sort of natural flow of things as if you were reading a book. And then if we look at the time series for this task, you can see here on the top is just the bold response. Proficient readers are shown in red and less proficient readers are shown in purple. You can see that the bold response actually looks very similar. In fact, if you compare bold, like proficient readers and non-proficient readers in this task, there aren't any differences in the brain. So typical block design analysis doesn't pull out any differences. But then if we look at the ISC, you can see very clearly here on the bottom part where the highly proficient readers show much higher levels of intersubject correlation. Particularly cool. We followed this up with a paper where Elizabeth Watts, who was a summer student here and then a special volunteer, took a similar experimental design, but instead of working with adolescents, these were emergent readers, so very young readers. And because they're very young readers who are just starting to read, instead of showing them stories and having them listen to stories, we just have them listen to stories. But we did have a series of like behavioral reading metrics and pre-reading metrics on them. So pretty similar as before. So 60-second blocks of listening to the story with breaks in between, and everyone heard 4 different stories. And if we look at our ISC of all participants, you can see here on the top 2 brains, again, we get nice language area in temporal lobes, particularly superior temporal lobes, expected and really cool. But we also see differences in the intersubject correlation in these, in like middle temporal gyrus that we weren't necessarily expecting. And again, I'll say, just like in the previous study with Dave Gingras' paper, if you just did a block design on this, there are no significant differences. Nothing comes out. But in the ISC analysis, it does. This ties in very nicely to the reading literature because we know that proficient and less proficient readers show differences in language areas, because reading is all about taking visual stimuli, absorbing it and pushing it as quickly and efficiently as possible into language cortex as you can. So this is a nice representation of single word reading studies in a more naturalistic way. But unlike Dave's study, where the sort of proficient readers always showed more connectivity, we had a more nuanced set of analyses that came out. So we actually had 4 different brain regions that came out where more proficient readers showed higher ISC than less proficient readers. But we had 2 brain regions that were actually more correlated in our less proficient readers than our proficient readers. This also fits nicely into the rest of the reading literature, which talks about compensatory mechanisms and how less proficient readers will often recruit additional cortex in order to process language, whether it's spoken, whether it's written and presented. The other thing that was particularly interesting is that just like Dave Gingras' study, we found that the Anna K model was very representative of certain brain areas. So again, language, auditory cortex, and even a little bit pushing up into angular gyrus and some frontal areas. But that wasn't the whole story. In fact, unlike the adolescent sample, in these children, we also had brain regions that responded to the different networking of nearest neighbors. So we had certain parts of cortex that said, hey, I have to get to a particular level of efficiency before I'm highly correlated to other people with that proficiency. And I had other parts of the brain that were like, hey, we read at about the same level. Our language scores are at about the same level, so we're going to be more correlated to each other. And it's this really exciting nuance of how you can have different brain regions that fit into each of these 2 classifications. And there's possibly even additional correlation structures that you could come up with that may fit this narrative and other narratives. And this is reading. I mean, reading is something that lots of studies have been done on. And we have a pretty good idea of how the brain reads in the scanner, particularly with single word. Gets replicated nicely here in naturalistic reading, which I thought was more fun. I think the participants thought was more fun. Yeah. Okay. A little bit ahead of schedule, I won't lie. I think I had more jokes planned. So obviously, research doesn't happen in void. So, special thanks to many people, including the co-authors on all these, or the leading authors on all of these papers, Emily, Dave, Elizabeth, of course, always thank my parents, my dog, and Sean Merritt. And with that, I will remind you that if you want more in-depth information about intersubject correlation, and particularly ISC and individual differences, please check out this workshop from 2021. Again, it is unparalleled. I feel like we'll run it again in the next couple of years, but yeah, it was a lot of fun. And there's also a few roundtables where we went into like, where is ISC going to go? And I rewatched them before the talk, and it's kind of fun because rewatching a roundtable from 3 years ago, people are like, it'd be really cool if someone would do this. And then you look at the publication since and you're like, hey, you said it would be really cool if someone did that. And then you went and did it. Way to go. Good job, science. And with that, I'll take any questions. A lot of things. Yeah.
Very cool talk. You very briefly mentioned the possibility of doing intersubject functional connectivity measures.
Mm-hmm.
Do you have any, or do you have any directions to point for someone who would be interested in a study like that? Like, are there examples that this has been used for scientific questions effectively?
Yep, there are a couple of papers that have used intersubject functional connectivity. I can point you in those directions. The one case that I always thought was particularly cool was hyperscanning, or any kind of, because my background is on reading and gambling tasks and addiction, was the like, can you drive other people's responses based on like, is there a strategy area and sort of a reaction area, and then how do those drive? And you could imagine that you could also have like these back and forth as you build these kind of networks to say like you were presented with this back from the other person, or like Mike Crowley has done these really cool studies called Cyberball, where people are playing catch. And so you're playing catch with these 2 imaginary people. And then at some point while doing this task, the other 2 people decide you're not cool enough. And so then they start playing catch with only each other. And so then you feel excluded. And so there's this question of like, okay, how does it feel like if you're the one being excluded? How does it feel like if you're the one excluding other people? Imagine that all of those would fit fairly well to sort of decision, action, and response. But those are my backgrounds. I'm sure if we had a competition online where people came up with cool studies, people would come up with some really cool ones. But I have the papers on my desk, so I can pass them to you. Other questions? Anyone online? Oh. Peter Kirk says, great talk, Pete. Do you think intersubject correlation is in nuisance parameters? For example, some subjects showing more similar movements than others are of significant concern. Should we be modeling all of these effects? Yes. I didn't go super into the weeds on how we get the time series for intersubject correlation, but realizing that I'm finishing like 10 minutes early, I probably could have. The first step in how I have done intersubject correlation is to run people through a pipeline that looks very similar to the AFNI resting state pipeline. So you end up putting the time series through effectively a regression where you're regressing out both the effects of the runs and also the effects of motion and the derivatives of motion. And then if you want to do things like anatocore, kinetocore, all the cores, you can do that. Motion is always going to be an issue, and it's always going to be something that you want to model. The cool feature that Bob Cox added to 3D Correlate when we started doing more ISC studies is that 3D Correlate can say, what voxel should I not add into the correlation between two subjects? And it could just be like, what volumes were censored? So if you have two time series going along, and for anyone who doesn't know, it's really rare in a time series to have an absolute 0.0. So if you come along in that time series and you have a 0 in one participant where they got censored for motion, and you don't have a 0 in the other participant where they didn't get censored for motion, 3D Correlate can exclude those TRs and say, I'm not going to put that into the eventual correlation. So your next question might be like, oh, what if you have someone with a lot of motion and someone who doesn't have very much motion? That's certainly a problem, and that's one of those quality control things that you should be checking to make sure that you don't have people who have like 10 TRs in their total movie watching and everyone else has like 10,000 TRs. These are quality control 101s. Any other questions? Yeah.
Can I ask a more maybe controversial question?
Yes, Tyler would like to ask a more controversial question.
So just thinking about it in relation to other types of multivariate analyses, it kind of seems like the mentality of ISC is a little bit at odds with things like hyperscanning, because at least in my mind, hyperscanning is kind of making an assumption that whatever is happening in one person's brain is also happening in a different person's brain, but the format is different. And ISC seems to be basically saying that things are all theoretically aligned, but they're just people deal with tasks in very different ways from each other. And that can be true sometimes or not true sometimes. Is that correct?
So yes, ISC generally assumes that everyone is having the same experience. Your previous question was on intersubject functional connectivity, so that's why I suggest the type of scanning. But yes, generally ISC assumes that people are doing or experiencing the same things with the same parameters.
But that those are separated in different brain areas. I'm thinking of the examples where you had results where you were saying, like, let's say Emily found that certain brain areas become correlated in a subset of individuals, but other people didn't have that portion of the experience or something like that. As where hyperalignment is more on the in line with like everyone's experience is somewhat the same. It's just that it's rotated or.
Oh, yeah. Actually, Fernando's paper that I reference goes deeply into why hyperalignment is critical is probably the right word. Critically essential is probably the better word for doing group studies, and ISC is particularly called out in that. Yeah, so that is one of the very fair critiques. I mean, it's an excellent paper, but it is one of the, I think, reading through it the first time, I was like, oh yeah, that makes complete sense. That if you have a different spatial representation of whatever you're doing in the brain, it may not be just voxel to voxel. It may be like, how do we non-linearly rotate or manipulate those things in hyperalignment in order to get them to line up to the same task in different people?
Sure. Okay.
Yeah.
And what happens when you don't?
What happens when you don't?
You don't align them correctly and you pass them through pipeline. Aligning is not the other part of the intervention.
Yeah. I mean, bad things generally happen.
Pretty things though. but-
Pretty things. I mean, one of the nice things about ISC is that, you know, your correlation, and again, most people have done fairly meaty ISC studies, right? Like fusiform face area makes a lot of sense. There are people who have done localizers beforehand in order to, like, functional localizers, and then try to use that functional localizer. So instead of doing full brain ISC, they'll do partial brain ISC. Some people will take parcellations of the brain, like the Shen Atlas, and they'll split that up, and then they'll use just the Shen Atlas ROIs and export all of those. So you're losing a lot of voxels, so to speak, but you are gaining perhaps the reassurance that it's not just like, oh, your fusiform area is like shifted a little bit this way, or like, oh, your I did a multilingual study recently. It's like, oh, your temporal pole is sensitive, but it's your dorsal temporal pole instead of this other person's like lateral temporal pole, things like that. But yeah, I agree. I think that registration is the thing that people continuously come back to in all of fMRI. And I am hopeful that more packages begin to offer different hyperalignment options. And hopefully Fernando's giving a talk on hyperalignment, because he's explained it to me at least twice. Every time it gets in there, into my brain just a little bit better. Any other questions online? Any questions in the room? Yeah.
That's a very basic question. So what is exactly the resting state at MRI? Like what state of a human brain state? Can I sleep? Can I think? Can I blink?
Well, yeah, so what is the state of someone in resting state? Yeah, that's the thing, is like, if your only task in resting state is to lay there and look at it cross, or like close your eyes, like we don't have a lot of control over if people fell asleep or didn't fall asleep. We may not even know if they fell asleep. Like, we have physiological measures. Everyone does resting state slightly different. I think every lab does resting state slightly different, because people read a paper and they were like, this is the one way I'm going to do it. But it does open lots of questions of like, what are people doing during resting state? And that's one of you don't have a lot of assumptions on resting state, but on the flip side, because you don't have any assumptions about like what their brain is doing, you also don't necessarily know what you're getting out, which is a con. But on the flip side, it's kind of awesome that you can pull out these 7+ resting state networks, no matter what people are doing. Like, so people have been doing resting state for differing varying amounts of time over like population after population. People have taken HCP or even UK Biobank and like stuck those in and said, hey, these networks exist, and they exist across everyone, regardless of what they're doing in the scanner. Yeah, sheer power of numbers and statistics. Yeah. Anybody else? See, I strategically left more time to talk for questions. We'll go with that. All right. Well, there's nothing else. We'll close this out. Happy Thursday, everyone.